Okay, so it's Thursday, the what, 22nd of May, and we're here in our fourth Biology 106 lecture for the semester. We've been on Chapter 12 now for a little while. Chapter 12 is a big one. It's an introduction to the nervous system, and we know a fair amount about the anatomy of the nervous system. That was much of what we've discussed in the past, and now we're still trying to figure out how in the world this thing works. It's really interesting. In lab, this week we discussed the senses. And I want you to think about this for a second. We know that nerves transmit electrical signals through these things called action potentials. And we're learning that every action potential is exactly the same, right? Every action potential is exactly the same. Now, different neurons are activated differently. For vision, photoreceptors, right? The light is what activates the signaling to be sent down the optic nerve. Or if we're dealing with the seventh or ninth nerve, those are chemoreceptors that trigger taste. Maybe sodium or sugar triggers the action potential to be sent to the brain. Or maybe we're dealing with mechanoreceptors in the semicircular canals for balance, or we're dealing with waves, sound waves moving the hair cells within the cochlea. Regardless, an action potential is sent, right? And so that action potential, that electrical signal is sent from somewhere in the body to another place. But it's always the same thing. It's always the same little negative 70 to negative 55 threshold to positive 35 and back down. Isn't it cool that our body can figure out all of this, even though the electrical signal traveling from one side to the other is the same? So now we have to start thinking, well, how is it then that the body can figure out that signal coming into the brain is visual information versus the information coming into the brain is taste or sound? Well, it has to do with where it started and where it ended in the brain. So as a simple example, we know that if light comes into the eye and the light triggers those photoreceptors found on the retina, that's going to cause action potentials to be sent through the optic nerve. Not all of those signals, but many of those signals are going to go where? To the occipital lobe, right? Where we call that the visual cortex, the part of the brain that receives that visual information. So how did the brain figure this out? Well, it knew where those signals were coming from, the eye, and it recognizes that when that signal arrives in a specialized area of the brain, hey, that must mean that that's visual information and not balance information or not taste information. So it's this amazingly complex thing that we're still trying to figure out. We don't have this completely figured out. But I hope you appreciate now that one neuron can be receiving signals from many other neurons, right? And this is the idea of summation, that one neuron can be receiving 10,000 signals simultaneously, especially if you're in the cerebellum, right, where you're receiving balance information. That's something that there's just thousands of signals being sent to your brain constantly. Now, how does a neuron do this? Signals can come in where? Either at the dendrite or at the soma, and yes, it also can come in at the axon terminals. It can come in and touch a neuron anywhere where there isn't myelin, created either by a Schwann cell or by oligodendrocytes in the central nervous system. And when those signals come in, they're going to be a local disturbance, a local stimulation on the neuron. We would call that a local potential. And that local potential, recall, is decremental. It gets weaker as it moves along. But what's one reason why it gets weaker? Why doesn't the action potential get weaker? Because there's myelin protecting the signal down the length of the axon. So there's no myelin in the soma. There's no myelin in the dendrite. So as a local disturbance occurs, that signal can kind of poop out as it moves toward the soma. We also learned that those signals coming in can be either excitatory or inhibitory. Now, I used that word initially, excitatory or inhibitory, and then as we were discussing last time, I added a different term to that, saying the signals could be EPSPs excitatory postsynaptic synapse or signals, or they could be inhibitory or negative signals. And what else was true about that signal? It was decremental. It decreased with time. It was reversible. It's possible basically to pull a local potential back or pull an EPSP back. And we saw that at the end of our conversation yesterday, because when another neuron comes in and synapses with an action, with an axon terminus, what can happen there? Let me go back up to that picture. Or maybe I have to go forward. Yeah, right here. Right? This is the situation where the big neuron, right? This coming down. This is the signal coming down, the axon terminus, and then we have the little synaptic knob, and the action potential is coming all the way down, right? An action potential is all or none, always goes all the way to the end of the axon. But oh look, there's another synaptic knob. There's another synapse happening over here. Now that particular neuron is right now doing nothing. It's turned off. It's not doing anything. And as a result, this action potential is able to come down and cause the release of those synaptic vesicles and the neurotransmitters released therein, and cause some sort of excitatory signal on the next neuron. But it's also possible for this neuron to come down and do what? Release negative neurotransmitters or inhibitory neurotransmitters. And one of those inhibitory neurotransmitters would be what? We discussed one, GABA. Whenever you hear the word GABA, you're thinking it is a inhibitory neurotransmitter. And what is it doing? It's basically overriding, isn't it? It's overriding the signal coming down the axon. Did the action potential make it to the end? Yes. But it was influenced by a greater number of negative signals or inhibitory signals coming from that other neuron, and that would be considered facilitation, or even more specifically, inhibition. Facilitator, what would you look for if this was truly facilitation? What would be different here? If this were facilitation, what would this neuron be releasing? Something that was excitatory, right? And would add to or facilitate or further increase the possibility of that signal firing. Here we see it's releasing, it says it's negative, right? It's an IPSP. So this would be a GABA-like neurotransmitter that is causing an inhibitory signal. Now, what is the molecular mechanism? How does a inhibitory neurotransmitter work? Let me back up. How does an excitatory neurotransmitter work? Typically, what we've seen is that the neurotransmitter touches the receptor, and as a result, a gate opens up, and that gate is what kind of gate? Sodium gate. Let's put a bigger word on that. If it opens up because a molecule has bound to that sodium gate, then we would more completely call that a ligand-gated sodium gate, because it was a ligand, a molecule that attached to it, that caused the gate to open and the sodium to go where? Into the cell, right? Which causes it to be more positive and causes a depolarization. If it were a negative or an inhibitory neurotransmitter, we talked about two different mechanisms as a possibility, and we found out that one of them was the reality. With GABA, what happens? GABA binds, but it doesn't bind to a sodium-gated channel. It binds to a chlorine-gated channel. And where is chlorine always in greater concentration? Inside or outside? Outside. And so when that GABA comes down and touches the GABA receptor, it causes the chlorine gates to open. Chlorine comes rushing in, and that makes that cell less likely to fire, right? It goes away from threshold. Is this all beginning to make sense? Okay, I know the people who, if it doesn't make sense to you, you're rarely likely to raise your hand and say, I don't get it. But I want you to do that if it's not making sense, because I want this to be a very important thing that you understand. Okay. So that's kind of where we were driving down the end toward this. I also want to go back to the idea of temporal versus spatial summation so that everyone understands this a little bit better. A dendrite rarely has just a neuron, sorry, rarely has just the 2 or 3 dendrites that cartoons show them to have. They would have hundreds to thousands of dendrites. And signals could be coming into those dendrites and on the surface of the soma from multiple places, and that's usually what's happening. So temporal summation was what idea? Who wants to share with me what temporal summation means to you? Temporal, when you hear tempo, you are thinking what? Time. So what's happening? Signals are coming down a single dendrite so rapidly, in such cessation of time, that in essence, there's a little bit of a stimulation, but the signals are coming so quickly that let me go to the next picture, that the neuron can't get back down to its resting membrane potential, right? So we continue to push a little bit closer and 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 a little bit closer because of that rapid firing of the signals until such time we do reach threshold. That would be temporal versus spatial. Spatial says that there are many signals coming in not from one but from multiple dendrites all simultaneously that basically do the same thing. Now, is this a one or the other phenomenon? I don't want you to think that neurons work one way or the other way. This is going on both systems, temporal and spatial, all the time. I'm just showing you 2 different mechanisms for this. But one axon is both receiving multiple signals down one dendrite as well as receiving multiple signals down multiple dendrites. Okay, so this is not one or the other. This is just 2 different ways by which these signals are added together to get closer to threshold. And where does threshold always happen? It's important that threshold occur where in the neuron? Yeah, we have to get a critical electrical signal to the axon hillock, right? Or what we would call the trigger zone. And at the trigger zone is where there's this massive number of what? Sodium channels. What kind of sodium channels are they here? Are they ligand gated or are they voltage gated? They're voltage gated because they open in response to a voltage disturbance. And then they fly open, and we know what happens at that point with an action potential. But again, down that axon, you've got myelin protecting in some way the axon, and so the signal is propagated all the way to the end. It's an all or none, never reversible. It's going to make it all the way to the end. Now, I did, in passing, mention a couple of diseases where the myelin breaks down, right? Where Tay-Sachs has some of that going on. MS more commonly has that going on. So, yes, the neurons fire, but the signal is not able to propagate down the axon smoothly. So these individuals start having some motor issues and some, what I always call hiccups in their giddy-ups. It just doesn't go smoothly, and their movements can become more problematic and more tiring in essence too. I haven't said it this semester yet, but myelin makes the action potential more efficient. It takes less energy, less ATP for your body to send an electrical signal down a myelinated axon than it does to send it down one that is not as myelinated. So when people have MS and they've got myelin issues, it literally is taking more energy from their body to get those signals to travel to the other end. Even though they're jerky and not smooth, it also is less efficient. So these individuals complain about being tired, right? They're wiped out. In part, I mean, it's not cause and effect, but in part because they are using more energy to do what for you and me with a normal myelin is not a problem.
So with the MS, does the deterioration of the myelin, does that affect the sodium channels in the node around here and the inner nodes? It just affects the sodium coming in with the electrical pulse?
I remember what I said, and you're on the right idea, Vince, and I don't know the complete answer to that. I want to see how far back I have to go to see that picture. I showed you, and it may be quite a ways back, where's the highest percentage of these sodium channels? At the axon hillock, and then again we see high densities of them Where? At the nodes around VA, and very few of them underneath the myelin. And another name for that area are the internode regions. And I think I said to you when I described that, I don't know what comes first. I don't know if the myelin, if the Schwann cell wraps around or the oligodendrocyte wraps around, and because it wraps around, fewer sodium gates are maintained there, or if somehow even before the myelin is laid down, there are these spaces where there are already a greater number of sodium channels. So I don't know which comes first. So then your question would be, okay, so we're losing myelin, and it's irregular, and so that means that there would be places where there aren't the high density of sodium channels like we would expect them. And so the action potentials don't travel as smoothly or as completely or at all down the axon. So in you and me, normal axon, it travels to the end because myelin's there. MS, other diseases where there's demyelination, then the signals are less smooth and maybe don't even reach the end. I know I didn't completely answer your question. I don't know the complete answer, but it's an interesting question. I don't know which comes first, the myelin or the sodium gates. Okay, so let's get moving ahead of here a little bit from where we were. Any questions up to this point? Is it beginning to make sense to you? Not just while I'm here talking to you, but are you able to turn to your neighbor and say, okay, I think I get this. Tell me if what I'm saying to you makes sense. And if you're not already doing that, I recommend that you do that, even if it's to yourself, right? Most of you have phones that have little cameras and video things on them and recorders. And I mean, if nothing else, turn on the recorder and explain to yourself in the mirror what an action potential is, and then replay it. And if it makes no sense, then you don't have it yet, right? So go back and study it again and then retell the story again. And you might go crazy doing that to yourself, because if you don't know it, how are you going to evaluate it? But it's always good, though, to talk with somebody else and make sure that what you're saying, the facts that you are putting into your sentences flow, that you're pulling the right information together. Okay, moving over past this idea of summation. And this is neurocoding. It's a way in which the nervous system is going to convert this information into a meaningful pathway or a meaningful pattern of action potentials. I've shown this to you. I just didn't use this word. So we can talk about how often a stimulus comes down a neuron and how often that receptor is being activated. And here's the idea. There's qualitative and there's quantitative. Qualitative would be how strong is the signal? Have I seen this signal before? And then quantitative would be what? How often, or maybe even stronger signals being sent down. Let me see if I can make more sense of this for you. How can the central nervous system judge the strength of an action potential? That's the question I want to ask you right now, and this will pull this together. When this action potential is traveling in an afferent neuron, a sensory neuron, and heading up your spinal cord and going to your brain, and it's received from the brain, what does the brain receive? It receives, if it's a single neuron and it's a single action potential, what's it getting? What kind of signal is it getting? It's the same thing over and over, right? An action potential is always the same. So the brain is receiving this little blip, right, up to positive 35 and back down to negative 70. Woohoo! How does the brain know if the signal it just sent was more important or less important? How does it know if it's something it has to respond to or not respond to? And this comes down to this integration of signals and to the coding of these signals coming in. So the bottom question here is, the CNS can judge the stimulus strength from how rapidly the axons are firing. So if it's only firing occasionally, then it's not a very strong signal. If instead you have something that's firing a lot, often, then that's going to tell the nervous system what? That this thing must be either more important or is a stronger signal. Look at these blebs on the screen here and figure this out. This is what the brain would see in essence when it was trying to lift something. Okay, so this is a signal that's going up to your brain, and you are voluntarily lifting an object. How do you know how many muscle fibers to recruit? So look at this. At 2 grams, what do you see? If you're lifting something that's only 2 grams, you're sending each of these little lines represents another action potential. They're not that often. Compared to if you're lifting 20 grams, you see how often those action potentials are firing. So the point is this: the more often the action potential is reaching the central nervous system, it tells the brain, right, that that's a greater intensity or a greater need. Let me keep working through this with you. There are also pathways of neurons. I alluded to you earlier that how does your eye know that when a signal comes to the brain that it's vision? Well, the brain knows that that pathway started back in the optic nerve. It knows that that signal came from the optic nerve and that many of those signals are ending up on the occipital lobe, right, for visual. This would be considered a pathway, and many of your neurons travel in groups, right, pathways or groups. And each of these pathways can have millions and millions of interneurons. Remind me, what is an interneuron? Those little neurons between the neurons. Where do we find interneurons? Completely contained within the CNS, right? So these are the little neurons that are firing within your spinal cord and within your brain. And many of these neuronal pools, or those neural pools, are going to help you do regular activities. When you start to walk, you initiate walking, right? You decide, I'm going to get up and I'm going to start walking. But does your brain have to continuously remind you how to walk? I mean, at some point it becomes automatic, doesn't it? It's not even something you're thinking about. And then when you stop, you also take control of it again. So you initiate it, but then you walk kind of almost involuntarily. You just kind of keep on going. And then at some point you make the conscious decision to stop, right? There are pathways within your brain. And through habituation, through doing things over and over and over and over, you can initiate it. The brain knows these signals, these synapses, these neurotransmitters that are being released, and then you have to stop it voluntarily as well. So these neural pools allow for one neuron to send signals to other neurons. And this is for repetitive things like breathing, right? It's a good thing, right? Our breathing patterns are regular. It's also something that happens when you're walking. Your limbs are moving rhythmically without any real voluntary thinking on your part. So information comes in at one of these neural pools and can branch. It will synapse with other neurons, and basically there's a pathway here that's very easy. When you're learning something new, it takes more work, doesn't it? Imagine a child learning how to ride a bike, or imagine, if you could, go back in time when your body was trying to figure out how to walk. And it takes a lot more work to get all that coordinated. But once you get it, right, it kind of happens automatically. Same idea here. Once those synapses are made and once those neurotransmitters are sort of, quote, trained to release more easily, then there are many repetitive activities that we do that don't require any true voluntary signal, although it is being controlled by the central nervous system. Now, these pools can work in zones, bigger zones, smaller zones, and I'm not going to get excited about this. So I'm just going to tell you, put a little gentle X through this. I'm not going to deal with this in any great deal. But I do want you to realize these pools come in different circuitries. And this is not new to you, but let me remind you that your neurons can work in diverging circuits. What does that mean to you? In a diverging circuit, that means that one neuron tells other neurons, which tell their friend neurons, and the signal is doing what? spreading out, right? It's diverging within your body. It's going to many places. Other circuits within your body are going to be converging circuits. What does that suggest? Many incoming signals perhaps are converging onto a single neuron, and that neuron might be an interneuron that is receiving thousands and thousands of signals simultaneously. There are also reverberating circuits, and this is what's going on with your highly repeatable things like your diaphragm and your intercostal muscles. Those things which are occurring with you in a rhythmic way constantly. When you hear the word reverberating, what comes to mind? Reverberating. Echo? Does the word echo come to mind? A reverberation or an echo? It's like it just kind of keeps going on and on and on and on. So a reverberating circuit is one where it's sort of cyclic and it starts a process over and over and over, or it's re-stimulated very, very easily. And then there are others that are called parallel after discharge circuits. And this is where the input neuron, the first one, diverges to stimulate several chains, and each chain has a different number of synapses which eventually come back and reconverge on the single one. And again, it kind of fires and fires and fires until it is stopped. We would never want to stop our diaphragm or our intercostal muscles for breathing, would we? So that reverberating pathway is kind of ongoing, ongoing, ongoing. The parallel after discharge, it goes until you basically tell it to stop, right? Or until the stimulus stops. So here are some pictures of these. I think they're easier to imagine in pictures. So diverging, one neuron tells all of its friends, and quickly, this is Facebook, right? Everyone knows. Number two, that was diverging. Converging, everybody, all these different neurons are converging, and they're sending their signals toward a limited number of neurons. Everything's converging at one neuron. When I look back at these, which two of these is more like the sympathetic nervous system, diverging or converging? Which one is more like the fight or flight response, sympathetic nervous system? Do you agree that when the bear walks in the room, all systems go on alert? Right? And so your sympathetic nervous system is considered very diverging because those signals are spreading to many places in your body very, very quickly. All point bulletin, everyone's alerted. Reverberating on the bottom, again, kind of this cycling, this echo chamber over and over and over. And then parallel after discharge, it's similar. You've got one neuron, it's speaking to multiple neurons, and they kind of go in a pathway until it stops. So just know it's reverberating, if you will, but then it has the ability to stop. And it's, just like it says, it goes parallel after it's discharged. So one signal is going to many places, but they're running along parallel after they are initiated. So again, we're just trying to figure out how does the nervous system pull all of these neurons together and make sense of it. You've got, trillions and trillions of neurons all trying, or trillions, billions and billions, I guess we should say, at any one time, trying to send signals and make sense of this. Let's go a little bit into one area of the brain related to memory. Okay, memory and plasticity. Somebody asked a question last time about plasticity, or they didn't say that word, but I use that word in my response. Plasticity is the ability of the brain to sort of recircuit. If something's been damaged, the brain is said to be plastic or have plasticity because it can overcome that injury and create new synapses or new connections, which oftentimes can go around that stroke or that area of problem, that area of damage within the brain. So let's figure out memory. How in the world does one hold on to memory? Every day you're making new memories. You are creating new memories. Your brain is keeping those or not. Right? We've got short-term memory. We've got long-term memory. We have the things that we can remember for just a short few seconds. Hopefully that's not this lecture, right? but parts of it is, right? Parts of what I'm saying are definitely short-term memory, and you're making sense of it as I say it, and then when you leave here, you're like, I don't know what he just said, right? Because you only got it as far as short-term memory, and you didn't capture it or create new memories from it. And then we have to take those short-term memory pieces and create longer-term memory and create new synapses. So let's figure out how this works. Memory is actually creating new synapses. Now, there's still some controversy, some discussion about this in neuroscience. Is every new memory that you make, a new phone number that you memorize, or whatever it is that you're memorizing, is every new fact another synapse? Or is it just a new way of using the pre-existing synapses? And some of that is still being debated in neuroscience, from what I understand. We don't have this figured out completely. Now, when you first start learning something, it's more difficult because you are creating new pathways. Even if we think about memories as being brand new synapses or brand new pathways, either way, it's the first time you've navigated through this path. So it always takes longer when you learn something the first time. And then the second and the third time, what happens? It gets easier and easier and becomes more second nature. And then eventually you've got this thing so well trained, this thought, that now you can think through it very, very quickly. Right? So for some people, math, right? It's more difficult for you. It takes you longer to process it. And other people who have mastered that are like, oh, nothing to it. Boom, they got it. They've actually created a more fluid pathway for that information. So we'll talk about immediate, short, and long-term memory. Okay, immediate, short, and long-term memory. Talk about the differences between them. And I'll go back and use this word again, synaptic plasticity. It's referring to the ability to change your synapses. Plastic, right? We can change the shape. We can remelt plastic and kind of change its shape. So when you hear about plasticity, think flexibility, changing the shape of something. And then potentiation. Potentiation is the idea that after a while, we make the transmission easier, that the signal travels more easily from neuron to neuron, and because it's traveling more easily, we say that the potentiation has increased. Okay? So let's take a look at first immediate memory. This is a phone number out of the phone book. Remember what a phone books are? Do anybody even use a phone book anymore? Right? They used to deliver them to the door, right? Anyway, but a phone book, right? You look it up and you'd have that number for a few seconds and you'd run to your phone and plug it in, right? That's immediate memory. This is why commercials repeat the darn phone number 17 times in 30 seconds, right? Because they want you to remember that number long enough to go to the phone. These are essential for reading, right? As you're reading, you're not memorizing that sentence, but that information is flowing into your brain. This is what we do when we're, again, just in the moment, not something we have to remember. Are some of you better with names than others? Some of you able to remember names, like you're at a gathering and you're meeting someone new. And for me, I'm horrible at that, unless I really think about it, right? If I just meet somebody new, I don't tend to remember their names. It's one of those, okay, in the moment things. Now, part of that might just be my vestibular deal that some of you will learn about later on. But I just don't remember names when I'm first introducing someone. I have to go back and practice it. I literally have to go back and think about it over and over and over and make that name stick in my head. And I do that with some of you on the way home. I'm like, okay. Now that name and that face are finally going together, and I kind of almost practice it. But by practicing it, now the next time I see you, I remember your name. Okay, or hopefully I remember your name, at least better than I used to. So those immediate memories, something you can't hold on to for more than just a few seconds. Then there's short-term memory. This is longer, a few seconds to maybe several hours. It's something you might quickly forget if you become distracted. This is even the phone number deal. You know, you might remember for a couple of hours, but then I remembered it yesterday. Now I don't remember it anymore. I knew it yesterday, now I don't know it anymore. That's still a short-term memory. Are you going to get it back? No, it's not going to suddenly come back usually. It's something that has come and gone. You held on to it for a short while, maybe as long as you needed it, and then when the next day comes back, you can't find it. That's a reverberating circuit. It's there, it's echoing for just a short while, and then it's, whoop, it's gone. Right? The echo dissipates. Now, facilitation, right? When I say facilitation, what comes to mind? One neuron helping another neuron, right? Do something, facilitating it. So we know that there is facilitation that can cause memories to hang in there longer. And part of this is a continual repetitive signal that comes at a synapse, a tetanic stimulation. And I haven't talked about calcium yet. I will be soon. Let me just say it now. When an action potential comes down to the synaptic knob, calcium must be involved to cause those synaptic vesicles to exocytose the neurotransmitters. I didn't show that yet in the pictures, and I will though. That as the action potential comes down toward the synaptic knob, there are calcium channels there. And those calcium channels are going to open and allow calcium to come into that environment, and that calcium is necessary for the exocytosis of the neurotransmitter. So calcium, if we continue to allow calcium to accumulate, we can actually have more synapses firing. And then we can also do some post-tetanic potentiation. That is sort of jog a memory. Sometimes you can jog a memory, right? You've had it, you've forgotten it, and then something else comes along that reminds you of it. Okay, we think that that also has to do with calcium levels at the synaptic knob. Long-term memory, a couple of different types, deal with things that you typically don't forget. We have declarative and procedural long-term memory. Well, declarative simply it means something you're going to put into words. I remember that, and I'm going to declare it. I'm going to put it into words. Procedural are things that you are now going to put into, quote, action. So if you have a long-term memory that's declarative, you can recall or declare that memory. You can retell the story. You can remember, oh, remember when we did that. That's declarative. Procedural is remembering how to ride a bike. Procedural is remembering, how to do the handstand that you haven't done in 10 years, but, oh, when I do it again, it seems to be working pretty well.
It would also be like, you have trouble spelling out a word, but when you write it down, it comes right when you spell the word or something.
Perhaps. What you're saying is, like when you're spelling, you're kind of mixing the declarative, right? You're beginning to spell, to remember it, but then as you physically go through muscle memory of your hand just kind of spells that word automatically as more of a procedural thing. Yes, right? And for some people, spelling is very much a motor thing, right? They may have a hard time remembering how to spell it ABC, but once they start to write it, it's almost like muscle memory. So for some of you you're not the best speller, one way around it is to go to the whiteboard and write something 20 times, make sure you're writing it the right way, and that you're getting a muscle memory going with the strokes. Vince?
So the procedural, that's just your muscle memory?
Pretty much. You can, retention of motor skills. Think of it as muscle memory. That'll be fine. Okay. Now, how do we maintain long-term memories? There is a physical rearranging of your synapses. There are new branches of axons, new synapses being made. And those molecular changes are collectively called long-term potentiation. In other words, you are now able to hold on to that memory long-term. And this could be decades. Okay? When a person starts having dementia, what do they normally forget first? The more recent things and more short-term memory things. But they might still remember their friends from high school or events that happened to them earlier in their life. But they typically start to lose the more recent memories. And then those long-term memories are the ones that stick around longer because that's just the way their brain's wired. Those synapses are not lost as quickly. Now, we know at the molecular level a little bit what's going on here. I told you that there's calcium involved. Well, there's also magnesium. Now, look at magnesium. Magnesium is a 2+ ion. What is calcium? Calcium's also 2+. Okay, so they share some similar ionic properties, some similar chemical properties. And here's the deal. Receptors on synaptic knobs are usually blocked by magnesium. Magnesium is there. Receptors on the synaptic knobs are usually blocked by magnesium ions. And when they bind glutamate, now glutamate, go back, glutamate, what kind of neurotransmitter was that? It's an amino acid, so it's an amino acid neurotransmitter. And did I tell you it was normally excitatory or inhibitory? Go back and look. Glutamate. Excitatory, right? What page is that on? Call out a page number, somebody? 33? Okay. So glutamate's excitatory. So when glutamate comes in, it repels the magnesium and allows the calcium to bind, and the calcium then allows more of these signals to go. And so basically, your long-term memory, I mean, it's amazing to think that memory comes down to a bunch of ions. But our entire nervous system comes down to a bunch of ions, doesn't it? It comes down to sodium and potassium and calcium, and in this case magnesium, all working in the right order to allow for these memories and these electrical signals to be transferring up and down our axons. So I'll talk a little bit more about some memory, and you'll see a couple of questions as well on the connect problems. So Alzheimer's, we can't open the newspaper without hearing about Alzheimer's. It's a really significant disease. Lots of deaths occurring from Alzheimer's every year. And, about 1/10 of the population over the age of 65 will be or has been diagnosed with some sort of Alzheimer-like changes. And what happens if they start losing memories for more recent events? They can become moody, they can become combative, not always, but oftentimes, and they eventually can lose the ability to do everyday things. Some speaking, some walking, talking, and they might even become dangerous because they'll pick up, for example, a steak knife and try to brush their teeth. Or they'll pick up, heaven forbid, they're in a garage with an electric saw, right? And they'll try to do something with the saw that is not very healthy. And so they just do these crazy things sometimes with tools. They forget how to use. That procedural memory is gone. Not only do they lose their declarative memory, they forget some of their memories, but they also lose their procedural memories. They lose how to do everyday activities. Okay. Now, we can't completely diagnose this, but we do know that there are some deficiencies in acetylcholine. Remember, acetylcholine was a most common neurotransmitter related to movement of muscles, but there's also a deficiency in nerve growth factor. We haven't talked about NGF before, but NGF, as its name suggests, is a factor that does what? Helps to grow new or help to maintain your nerves. So if you have a decrease in NGF, then your nervous system wouldn't be able to maintain or create new memories or new synapses or new nerves. And upon autopsy, what you'll see is that there's an atrophy to the gyri, right? The brain is shrunken. There's an atrophy to those gyruses. Remember, the gyri are in the cortex, right, the outer layer of the brain where all that gray matter is. And also, you'll see a buildup of plaques. So this is an amyloid product, and it builds up and sort of tangles up and messes with the communication within the central nervous system. Not a lot we can do. There are some medications right now that are trying to halt that beta amyloid production, trying to slow down the plaque production. There are also some medications out there now trying to give nerve growth factor to, the drugs you're hearing about now. What's the newest one in the market? Something rest. Something. And there's a couple of them out there that you'll hear about that are trying to slow down the progression of Alzheimer's. So if it's diagnosed early enough, they can give them some meds. So here's a brain, and you can kind of see, I don't have a normal right next to it, but you can see that on a normal brain, you would not have these big gaping holes between, right? The sulci would be really small. So you can kind of appreciate that these gyri, the folds have shrunken, making larger spaces between them. And then if you were to look under the microscope, you would see the accumulation of these plaques. And they're basically, again, just an amyloid, a protein that accumulates that begins to, quote, tangle up or create problems with the nervous system. Another disease of the brain that we hear a lot about is Parkinson's disease. This is a progressive loss of motor control. It begins typically in one's 50s or 60s. There is no recovery or really good effective treatment, although there are some things we can do to slow it down. We know that this has to do with the degeneration of dopamine-releasing neurons from an area of the brain called the basal nucleus, or the basal nuclei, plural. The basal nucleus is on your list, the functional areas, okay, on the brain. As I was leaving Tuesday, I was saying something to somebody that I think everyone needs to hear again. Every neuron in your body can only make one neurotransmitter. Okay? So when the action potential comes down a neuron and it releases a neurotransmitter, whatever kind of neuron it is, it's going to release that particular kind of neurotransmitter. Remember, if it's a cholinergic neuron, it's going to release acetylcholine. If it's an adrenergic neuron, it was going to release norepinephrine. If it was a GABAergic neuron, it was going to release GABA, and the list goes on. In other words, a neuron doesn't sit there with 100 different kinds of neurotransmitters in its synaptic knob and then somehow decide which neurotransmitters to release. It's all or none, right? So a neuron only has one job to do. It only releases one type of neurotransmitter when it is stimulated. So in the basal nucleus, there are neurons that release dopamine. So we would say that they are dopanergic, right? They're dopanergic neurons. And their job is to release dopamine. Well, dopamine goes on to control much of your motor activity. In fact, dopamine inhibits motor movement. So read this statement. Dopamine normally prevents excessive activity. So if you lose the dopamine, if the basal nucleus is no longer making dopamine, you have lost a level of inhibition. You've lost a level of control over the fluidity of your motor movements. So a person with Parkinson's from this area of the brain called the basal nucleus no longer is producing enough dopamine. And as a result, the dopamine no longer is inhibiting excessive motor movement. So what are the symptoms of Parkinson's? Excessive motion, right? And so these individuals start getting these Parkinsonian tremors. If they had normal dopamine, then that excessive movement would be restricted again, right? So because they don't have the dopamine, they're shaking uncontrollably. Okay, so not much we can do. We're getting better at it. We can give people a drug that's dopamine-like. The problem with it is that this drug has to get up into the central nervous system, and the blood-brain barrier is pretty strong and doesn't allow dopamine. You can't just give somebody dopamine and get it to the brain to replace what was being lost. So we have to give them another medication. The problem is the side effects are pretty crappy. And so people have to choose between Parkinsonian tremors or the crappy side effects of trying to control it. And a lot of folks will, for a while, do the meds, trying to keep their Parkinson's under control until the side effects become so overwhelming that they say, forget it, and they go back to doing their tremors. And so it's kind of this constant ebbing and flowing of trying to control it and not trying to control it. There's also some surgical things that are being done to try to control that. Okay, so that brings us finally to the end of chapter 12. We've been doing this for 2 lectures plus, haven't we? It's been a large section. This chapter will be, I'd say probably a good third of your exam. And remember that on Blackboard, let me go there now with you. If you go under lecture materials and go under exam 1, you may not have looked at it yet, but down here underneath the PowerPoints, it says guided reading number 1, and this covers chapters 12 and 24. So open that document up, and it's just a Word document. Go to the library and use your free copies there to print this. Now, have I discussed everything in here? No. Okay, right now I'll tell you there are some things in here I've not discussed necessarily, but a lot of this is just a list of vocabulary terms. And if I have mentioned it, then you want to make sure you know how to use that word, or if it were in a question, that you would recognize. Remember, there was sensible and insensible water loss, and I mentioned obligatory water loss. Those were chapter 24 concepts. Then here in chapter 12, there's quite a few terms, right? And then after that, you'll find some short answer questions. And these are questions that would help you formulate your ideas, okay? And there's quite a few of them. I don't collect this, but if you were to ask me for a study guide, I would send you here. Okay. And it tells you what chapters and Saladin to read and what sections, what pages to look at. So take a look at those documents. I think you'll find them to be helpful. Again, I won't be collecting them, so you don't have to do them in perfect form or plan on passing them in or anything like that. Check those out. So between the guided readings and your online Connect assignments and your quizzes, right, you should have lots of practice in being prepared for your exam. That exam is still a ways off, right? It's still a week from this coming Tuesday, June 3rd. We've got a holiday weekend coming up and then another whole week before that exam. So it's gonna come up faster than what we anticipate. So don't wait until after Memorial Day weekend to begin to pull some of these ideas together. I would beg you not to do that. So it's going to be chapter 24 on the water balance, chapter 12, neurons and all this, and then chapter 14. Now, there's not a whole lot here. This is a relatively short chapter. I'm pretty confident I'll finish this content here today. And this is going to go into sleep and into parts of the brain. Now, in lab, you have that functional anatomy list, that long list of structures. What's that lobe doing? What's that part of the brain doing? What's that nucleus doing? And almost every one of those structures I'm going to mention here in the next hour or so. So you're going to have a better understanding of why I had you learn those parts of the brain. Okay, let me pause there for a second. Anything else so you can think of related to chapter 12 to clarify? Okay, so let's get going with chapter 14. We finished chapter 12. Is there anything at all? Anything at all? It seems like we've just got to look through our Connect assignments and look through those guided reading assignments and determine what is important to me. And then I'll give you some more clues next week. Don't forget, too, that there are 3 quizzes associated with this first exam. And you'll find them in Connect. And they're going to be the first quiz. Anybody recall right offhand? The first quiz, they're all due at the same time, right? And they're all due on the 1st. The exam is on the 3rd. But the quizzes are due the day or so before that. That was, yeah, I was just hiding yours. No, it was my bad. So right here, lecture quiz 1, 2, and 3, right? And they're all due on the 1st at midnight. So that's Sunday night. Sunday night. And so that's a week from this, that's 9 days from now. And they're all due at that point. One is on fluid, chapter 24. The second quiz is on nervous system. That's pretty much what we just finished up. And then there'll be some here on the senses. This is chapter 16. Chapter 16. So each of those will help guide you as well. And then as soon as you're done with those, at 12:01 a.m. on the 2nd, Monday morning, those quizzes will be available for review. So you'll have Monday and Tuesday to review those quizzes. So all that should hopefully guide you to being very successful in this first test.
How many questions?
Oh, about 100 and something questions. Maybe, I don't know. We've got all 2 hours to take the exam, so there's no shortage of time. Yes, ma'am.
It's not timed, right?
Well, the quizzes, no. No, the quizzes are not timed. The exam will be the period that we're together, the 2 hours, but no, the quizzes are not timed.
And only one entry though.
One, you can come and go. You can change your answers. Only one submit.
Okay. So I can say.
You can go to the next question and then go back and change your answer. Okay, so they're completely user-friendly. You're not forced to go to the next question. You can come and go back, but only submit once. Now, if you forget to submit, it will submit for you at midnight on June 1st. So if you've done only 3 of the questions, it will submit it for you as completed at that time. So if you forget to submit it, we'll submit it for you. But only one submission.
Are those graded? Are those graded like they were last semester?
They're each worth 10 points.
Yeah.
Yep, and they'll go into your grade book as 10 points. Okay, so let's get into chapter 14 a little bit here. We're talking about some sleep. We all need some. And some higher cognitive functioning ideas within the brain. So the first thing we're going to deal with is sleep. And let me give you a couple of structures here before we get there. The basal nuclei, it's plural, right? Nucleus, nuclei, plural. Now, what in the brain is a nucleus? Let's go back to that. What is a nucleus when you're discussing neuroscience? Cluster of cell bodies found within the central nervous system. I'm not talking about an atomic nucleus where the protons are. I'm not talking about a cellular nucleus where the DNA is found. The nucleus here is a cluster of cell bodies found somewhere within the central nervous system. Now, what color would cell bodies be? Gray or white? Gray, because cell bodies would not have around them any myelin. So cell bodies, a cluster of cell bodies would by just appearance be gray. Anything that has myelin around it, aka axons, would appear white. So what we have when someone slices into the brain, just imagine this, throughout years, people have been slicing through brains, and the majority of the brain is white matter. But there are areas of gray matter. And some of them are these little islands, clusters, and so they all have a name. So there are the basal nuclei. Now, looking at this section through the brain, it turns out that there are 3 centers that make up this thing called the basal nuclei. And you saw them all on your list from the lab. There's the caudate nucleus, there's the putamen, and the globus pallidus. Now, they're all labeled here for us. I know it's hard to see this. The putamen is this green area. The globus pallidus is this green area here. And then, where's my last one? There it is. The caudate nucleus is up here, this brown one. Those 3 areas are collectively the basal nuclei. There's a left and a right. The big purple area you're seeing there in the center is the thalamus. Remember, the thalamus is in the very center, the nugget, the core, the pit of the brain, and these basal nuclei are just out of that. Now, what are they doing? They are receiving collectively, these nuclei are receiving input from an area called the substantia nigra and from the motor areas of the cortex. So collectively, they are involved in motor control. So you want to have that as your key idea, that these basal nuclei are part of your motor control. Now, are they in your involuntary or voluntary portion of your brain? They're down by the thalamus. They are part of your involuntary portion. So they're helping to coordinate your overall movement. And a moment ago, I mentioned substantia nigra for what? Go back to Parkinson's. Did I mention the word substantia nigra back in Parkinson's? Or not?
No.
No? Okay. But that's also speaking in that area, okay? So that whole idea of motor control and fluidity of your motion, that's all informing your overall motor control. So those are the basal nuclei. From Anatomy and Physiology Revealed, if you were to search for the basal nuclei within that program, remember that's the program that's linked off from Connect for you. It is a cadaver dissection tool, and you would see these three areas. I'm going to tell you right now, I'm not going to have you differentiate between these three. Okay. On the lab exam or on the lecture exam, I would identify these areas for you, and I would more want you to know, what do the basal nuclei do? Right? I'm not going to say, oh, is that the putamen or is that the globus pallidus? But I would want you to know that collectively these basal nuclei are involved in motor control. It's your involuntary side of it, right? It's the coordination of your motor control. Another part of the brain that I want you to know before we get to sleep is the limbic system. Now, whenever you hear the word system, you're thinking, oh, we've talked about the urinary system and the respiratory system and the musculoskeletal systems, and I kind of wish they didn't have this word here, system. It's not one of our body systems, but it is a series of structures found within the brain that are all involved with your emotional centers. So when you hear the limbic system, I want you thinking, okay, this is the area of your brain involved in your emotions. It also has a fair amount to do with your overall learning. Okay. So and this makes sense if I just say it immediately. If someone is having emotional distress in their life, how's their learning doing? Right? So clearly these things are connected, right? So if you're emotionally, quote, unstable, then you're not going to be able to make the same learning outcomes as you would if you weren't quite as emotionally stressed out. Now, within this limbic system, within this series of structures, you will find the cingulate gyrus. Now, what's a gyrus? One of those folds, right, the noodles of the brain. And this particular noodle of the brain is the one that goes right over the corpus callosum. So you can't miss it, right? You all can recognize the corpus callosum. It's always a paler, white structure. And what I'm coloring in right now is the corpus, right? And that blue fold right above it, that's the cingulate gyrus. It arches over the corpus callosum. It's in the frontal and the parietal lobes. There's also, as a part of the limbic system, the hippocampus and the amygdala. They were the last two terms. Hippocampus and amygdala were the last two terms on that functional anatomy list in the lab. They were on the front page. And then when you go to the back page, I believe that was the basal nucleus, right? So the putamen, the caudate, those structures were on the back side of that page. So I'm trying to show you that all of those structures in that lab list we're going to discuss here in some way. Now the hippocampus, when you hear the hippocampus, I want you thinking memory. Okay, within the limbic system, the hippocampus is a lot to do with your memory, whereas the amygdala is your emotion. Collectively, they make up a large part of your limbic system, which is both emotions and memory, right, or emotions and learning. So if you hear the word hippocampus, I want you to immediately think memory. When you hear the word amygdala, I want you to immediately think your emotions. Now, where are we here? This limbic system is in what would we call this region of the brain? Overall, that region of the brain is the which encephalon? The diencephalon. And what did that if you remember that word from 105, what that means is that's the pit, the core, the nugget, the center of the brain near the thalamus. And we're still in some of that involuntary part of the brain. We're not up in the cortex where we're dealing with our voluntary choices. And there's a lot of information going right through the center of the brain. So your memory and your learning and your emotions are right in the middle of all of these axons traveling up and down. The limbic system is also the center for your gratification and your aversion. So the parts of the brain that addicts have, there's a center of a gratification that is triggered through some drugs and activities, and that sense of gratification that the addicts need to deal with is focused here within the limbic system, as well as areas of fear or sorrow or aversion. So sometimes we're drawn toward things for gratification, other times we're drawn away or pushed away from things because of aversion, and all of that is part of your limbic system. Okay, so just think limbic system, emotions, learning, amygdala, emotions, hippocampus, memory. And then let's go into the other functions of the brain. Some of you were frustrated when you were looking at Google or your textbook and trying to nail down one little thing that some of those regions of your brain did. If I say to you frontal lobe, many of you will tell me motor, but you'll find other things that the frontal lobe is responsible for. Or if I said temporal lobe, you're not going to find just one thing. And some of you are kind of frustrated because you want that one and only answer. You want that black and white. If I memorize this, I'm okay. And you will be, but I hope you realize that there are many parts of the brain that integrate together to give you your overall well-being. And many of these are considered the higher brain functions or your higher cognitive functions. So this is going to include some of your higher brain functions would integrate many areas, and this would be your sleep, memory, cognition, your remembrance of things, your emotions, sensations, control, language. Now, that's not to say that my daughter's dog doesn't have a form of language, right? It can bark and it can whine, but its language skills are certainly not what you or me have. And yes, the dog has wonderful motor control and it does sleep, and I'm sure it remembers when, its name is called or it needs food, but it's not the same, is it? Right, as what we have in our phenomenal brain. Now, these integrated functions of the brain are going to involve not only your cerebral cortex, and when I say cerebral cortex, I want you thinking that's the part of the brain that's all voluntary, right? This is the outer layer of your brain where you are consciously aware of what's going on. But it's not only your consciousness, but it's also an interaction with your basal nuclei, your brainstem, and your cerebellum. And we recognize those are all areas of your brain that are involuntary, over which we have no control. So you've got this integration going on between these areas of your brain. And like I said, it's hard to say that what does this little area of your brain do because there is such an integration. This is also good news, though, because if you do suffer a stroke, if you suffer an area of your brain, there's a chance, right, if it's not too big of an area, that other parts of your brain will sort of, because of their plasticity, other parts of the brain will come along and sort of compensate or overcome for that deficit. To a point, right? To a point. So we see that it's hard to identify one area of the brain that does something sometimes.
It also depends on age.
Oh, the age and your ability to recover from it and how extensive the damage was and exactly which areas, because some parts of your brain are exclusive. I mean, there are things that only occur in a small part of your brain. And if you lost that part, it's not likely that another part can overlap and compensate for it. So it all depends upon exactly where, too, that damage occurred. Okay, let's get back to sleep. We're going to learn a little bit about sleep here. Now, in this semester, we will be adding electrodes to you, and we will be measuring the electrical activity of your muscles. We'll be doing that in about 2 weeks in lab. We'll be doing what we call EMGs, electromyograms. And then later in the course, we'll be doing ECGs or EKGs, electrocardiograms. So muscles send off electrical signals. Heart, muscle sends off electrical signals. Remember that muscle is excitable. But the nervous system is also excitable. So your brain is also sending out electrical signals. And these are picked up by electrodes. And these signals are called your EEGs or your electroencephalogram, right? Well, we can measure these electrical waves coming from your brain. If someone has no brain waves, we say that they are clinically dead or brain dead, even if their heart is still beating. If they don't have these waves, then we've determined that they're in some sort of vegetative state or completely brain dead, right? So we can hook up electrodes to the forehead, to the scalp, and determine the electrical activity of the brain, and it will help us to determine if things are okay or not. There are normal waves that we should expect to be released from your brain while you're awake, others that are more common when your eyes are closed and you're relaxed, other waves that are present more when you are sleeping than when you're awake. So we'll look at these different waves, and if there's an abnormal collection of these waves, then maybe you have some sort of epilepsy, or maybe you have a sleep disorder, or maybe you have even a brain tumor of sorts, or some other degenerative brain disease. So again, these brain waves are cyclic. They come and they go. They're rhythmic voltage changes, and they're coming from the cortex. So they're coming from the outer layer of your brain, where all those synapses are being made as things come up the spinal cord and past the thalamus and into your cortex. We're measuring the collection of all of those electrical stimulations. They're going to be measured in millivolts. We've seen millivolts already, right? -70, -55, +35. Millivolts were our unit of the voltage, and that's the amplitude, if you will. And then how often the waves are coming in, that's their frequency. And frequency is measured in units of hertz, Hz. Again, persistent absence of these waves would suggest a clinical or legal definition of death. So here are the waves. The alpha, beta, theta, and delta waves. Okay? Let's figure out when they're there and what's going on. Alpha waves, 8 to 13 hertz. That means that's how often they're occurring. Okay? And these waves are going to be most visible on an EEG when you're awake, your eyes are closed, you're just kind of resting your eyes, and you are just kind of thinking about nothing in particular. If you were to begin to think about some particular mental task, these are typically suppressed. So if you just got you're awake, your eyes are closed, and you're thinking about nothing in particular, you would have alpha waves. The moment you began to concentrate on something, or the moment you opened your eyes, those alpha waves would go away. Beta waves are going to be picked up then. The beta waves are when your eyes are open or when you're mentally challenging doing something. So if you were being asked to do a bunch of mental, multiplication problems or something like that, then you would expect to see, with your eyes open and doing mental math, you would expect to see the beta waves pick up. They are especially accentuated when you are really thinking about something hard. Theta waves, maybe some of you are experiencing them right now. You're getting drowsy, right? And your theta waves are going to kick up. And they also can be present if you are under a lot of stress. And then finally, the delta waves are in deep sleep. So that's how I remember that one, the DD. The delta waves are during deep sleep. They have the highest amplitude. They're the biggest waves, but they're not that often, right? 3 to 3.5 hertz. So they're not as often, but they're the largest of the waves. Let me put this into more information, then we'll look at some pictures of this. Now, when you're sleeping, and if you've taken who's had psych? And if you've had psych, you've heard this story, maybe from a different perspective, but you've heard about REM sleep, and you know that we go through these sleep patterns while we sleep. So during sleep, there are these cycles of these patterns. They're largely based upon our circadian rhythms. Circadian rhythms are the idea that we have a 24-hour clock within us. We're not sure, but we think the pineal gland, right, has a lot to do with sort of regulating and setting that 24-hour clock. And sleep is that welcome temporary lack of consciousness, right? It's that time when we're no longer conscious, but we can be awoken if stimulated. Now, typically, we're sleeping, we're laying down, our eyes are closed. During this time, there is a paralysis, a reasonable paralysis, where your muscles are actually inhibited so that you're not banging, your partner or banging your hand all over the place and bruising your arm. And your sleep resembles unconsciousness, except you can't be aroused from unconsciousness. You can be aroused from sleep. If instead you are in a prolonged unconscious state, you are in a coma, or you're a bear. Okay? You're hibernating. Okay? So why do we need sleep? As she yawns. Why do we need sleep? We need some right now. Why do we need sleep? It's a time during which our body can regenerate its store of energy. It's a time of parasympathetic, where rest and digest is occurring. Our brain is restoring its glycogen levels, which are the energy storage molecules. Our ATP levels are being restored. And this is especially a time when your memories are strengthened. This is why I always encourage you, if I haven't said it this semester to this group, I always encourage you, before you go to sleep tonight, review today's notes. Because you will form memories while you are sleeping. And those new synapses and some of that potentiation and some of that, long or short to long-term memory happens when you are sleeping. And if you don't have anything there to create new memories, then nothing's going to happen. So while this story is fresher in your mind, and while this conversation, this lecture is fresher, it's better to review your notes and then go to sleep and let some of those memories firm up. The day of a test, we all fall asleep studying, right? The day before a test, right? You fall asleep and you wake up, and the moment, 3 hours, 4 hours, however many hours you sleep, 6 hours later, all of a sudden you wake up and you seem like you're right where you left off. You're already thinking about what you think you fell asleep 6 hours ago, and all of a sudden it's right there on the tip of your tongue, right? So you're creating some of those memory connections during sleep. So we need to have it. We need to have this sleep. Now, there's an REM and non-REM sleep, and I'll get into that in a second. So there's 4 stages of sleep. The first stage, you're getting drowsy, right? Your eyes are closing. You're beginning to relax. You may feel like you're kind of drifting off a little bit. You'd be easily awoken. This is me falling asleep in my recliner once in a while, right? I just barely fell asleep, and I can easily be awoken. During this time, what kind of waves would I have? My eyes are closed, and I'm drifting, so that would be alpha waves, right? Stage 2, passing into a lighter sleep. The EEG levels are going to start to decline in frequency, but increase in the wave size. So the waves won't be as often, but the waves will be larger in their amplitude. And during this time, there'll also be some spikes going on, some electrical spikes. And we know that that's the thalamus talking with the cerebral cortex. So you've got this increased electrical activity spiking in your brain. Stage 3, you're in a moderate to deep sleep. This is going to occur on average about 20 minutes after stage 1. At this point, your theta and delta waves are going to appear. And your muscles will be at this point the most relaxed, and your vital signs will be at their most relaxed. So your heart rate's slowest, your breathing is the slowest. All of those things have fallen to their lowest levels. And then there's stage 4. And stage 4 is called a slow wave sleep. And this is dominated by, I told you this would be the what waves? Delta and the deep sleep. And during the delta waves, they aren't very common, they're not very often, but when they do occur, they are huge, right? High amplitude, big waves. At this point, again, your muscles are very relaxed, your vital signs are at their very low, and this is the time where it's most difficult to wake you up. However, during that night, go ahead.
What stage is it when you feel like you're you're almost asleep and you feel like you're gonna fall off something, you like twitch and wake up?
I'd say you're in between 1 and 2. Because once you're into 3, you wouldn't be just spontaneously waking up, right? So that you start to fall asleep, and all of a sudden you feel like you're falling, or that little, perk up. That's between 1 and 2. Okay. Now, during the night, if you get enough sleep, during the night, on average, around 5 times, a sleeper will backtrack from stages 3 and 4 and go back to stage 2. And during this time of going backwards, they will exhibit rapid eye movements. And so this is referred to as rapid eye sleep, or REM sleep. And during this time, the eyes will oscillate back and forth. It's always fun to watch a dog in this stage, right? Right? Their eyes are moving around, and they're kind of whining and making noises sometimes a little bit. Now, this is called paradoxical sleep as well, because the EEG is going to look like you're awake. The EEG is going to increase its activity, but you are actually still in a deep sleep. In fact, you're in a sleep deeper that you're more difficult to arouse. So you're physically you're more difficult to arouse, but your brain activity is increasing. And during this REM sleep, not only are your eyes moving around, but your vital signs increase. Your brain is using more oxygen. And but also during this time, you are going to have a greater muscle paralysis, so that you're not going to be acting out these dreams or whatever it is that this increased activity is being caused by. Now, you can have dreams during either REM or non-REM sleep. People who have dreams during their REM sleep will say that they have longer, more vivid dreams. Who here has vivid dreams? Anybody not dream at all? I haven't had a dream in, I don't remember having a dream, 30 years? I don't have any recollection of any dreams. If that means something bad, let me know privately. I don't know. But I truly wake up and, okay, it's just another day, but I cannot remember. It's been years and years. As a kid, I remember having dreams. But as an adult, I honestly don't remember having any good, bad, ugly, indifferent, nothing. And then I have these friends who say, oh, I had a vivid dream. It was in full color and this whole thing. And I'm like, I don't know what you're talking about. I just can't appreciate that. So am I alone? Am I the only non-dreamer? Wow. Are they good? Scary? Sometimes scary, sometimes just bizarre? Maybe I've replaced my sleep, my dreaming with my ride back and forth to Grand Rapids. Maybe that's my time to doze off. I don't know. Maybe that's my brain has said you've had enough entertainment for the day.
What about like night terrors, like kids?
Yeah, well, it's a nightmare, right? It's some sort of...
They can't.
They can't what?
They don't wake up out of it.
Right, they don't wake up, right? So they just kind of, my little guy, once in a while, he'll have a little night terror thing. He's screaming, but he's not awake. And he doesn't seem to be aware of what's going on. So I don't know. Yeah.
Is that in REM sleep?
I would think it'd be during REM sleep because it's the most difficult to wake them up. But clearly they're having some sort of brain stimulus, right? They're upset about something.
But isn't the paralysis supposed to be deeper in REM sleep?
It is. My guy he just screams. He's not moving. Yeah.
What about sleepwalking?
Yeah, sleepwalking wouldn't be this deep paralysis, would it? It wouldn't be during this deep paralysis. Perhaps stage 3. Yeah. Now, during REM sleep, your parasympathetic nervous system is also heightened, that rest and digest period. And this is a time where if you were to look at the pupils, they would be constricted, right? Do you agree that constriction of the pupils is parasympathetic? When you're scared, sympathetic, the pupils go, woo, big. So when you're relaxed, right, parasympathetic, the constriction of the pupils. And this would also be a time of erection, not related to any sort of stimulation. So sleep then, this rhythm of sleep is controlled by all sorts of things. An interaction, again, between your cerebral cortex, the thalamus, the hypothalamus, the reticular formation. Where's that reticular formation? It's all the axons going up and down through the brainstem, right, the medulla oblongata. And arousal is induced in the upper reticular formation up by the pons and in the medulla. And sleep, we know, is induced below the pons, a little bit deeper and in a section of the hypothalamus. I did not have you know the ventrolateral preoptic nucleus, so don't worry about it. I didn't ask you that in lab. Now here's what I did mention in lab, the suprachiasmatic nucleus, right, the SCN. It's an important control center for sleep. So as you're going back and looking at that functional region of the brain, worksheet in lab, and you get to the SCN, put down next to it a control center for sleep. Now, its name should tell you something. What does supra mean? Above, and chiasm or chiasma is referring to the crossing over. The chi is the Greek letter for X, or X is the, how's that go? Chi is Greek letter X, right? So the crossing over the optic nerve is called the chiasm. So it's right above the optic chiasm, suprachiasmatic nucleus. It's a cluster of cell bodies found right there. And again, they're involved with sleep, control center for sleep, and they help with our circadian rhythms and some of our other 24-hour clock type things. Now, we've already said that sleep is necessary to restore your energy levels, and a lack of sleep is fatal in experimental animals. Perhaps fatal in you and me too, right? But we don't get to do those experiments anymore. You know, ethics gets in the way. I almost said that's what Guantanamo's for, but I don't think so. I didn't say that out loud, did I? So we know that it takes more. The bed rest alone does not have the same. Bed rest alone does not have the same restorative effect as sleep. So we know that just laying down is not the same as getting a good night's sleep. Maybe some of you are a cat sleeper. You just kind of take little naps here and there. And after a while, that isn't quite the same usually as getting a good night's sleep occasionally. And we've already mentioned that we need REM sleep to consolidate our memories, but also to restore our energy levels. So let's look at a chart here of the sleep stages. So, and also on here are those EEG waves. So in the first one, you're awake. And this is, you're just awake, right? So what waves might you see? If your eyes are open and you're thinking through things, you'd be seeing beta waves, right? And then you go into stage 1. Now, these minutes are down here. It's kind of an average. But you go into stage 1, so you just close your eyes. You're getting drowsy. You're kind of drifting off. You'd now be having alpha waves. I wish they were in order, but they're not, right? So beta waves when you're out and around, eyes open, concentrating, doing things. Close your eyes, get drowsy, alpha waves. And then a little bit later, we get to a deeper sleep. Now, I told you that during that deeper sleep, you'll start getting some spindles. And they're not as obvious here as I'd like them to be, but just jumps, bigger jumps in the waves. And that's the thalamus talking with the cortex and doing some electrical activity. And then you'll get to that stage 3, that moderate to deep sleep, and eventually to your deepest sleep. And what kind of waves are those? Those are the delta waves, right? But then what happens is that there are going to be these periodic times where you come out of deep sleep and return to what's equivalent to like stage 2 or so. And this is the REM sleep, okay, the REM sleep. Now that's showing one stage, and this is showing that it's happening somewhere about an hour or so after sleep. In the progression of a long night's sleep, you would go through, this is a typical 8-hour sleep period. Who gets 8 hours of sleep? Anyone? Wow. Good for you, okay? Most of us didn't raise our hands, did we? Okay? So one of the reasons they say that we need to have an 8-hour sleep or 7-hour sleep is that we get enough REM cycles within that sleep. So if you're just sleeping an hour at a time, you're never really going to get into REM. You're not going to have that restorative time, and you're not going to have those memories being formed unless you go through these REM cycles.
Why is it that some people specifically can only sleep up to like 6 or 7 hours?
Yeah, some people really, their little clock doesn't work, right? They just can't sleep in. I mean, just sleeping into them is 5 a.m., right? Anybody have grandparents like that? I mean, I don't know. It just seems to be the elderly. All of a sudden, it doesn't matter what time they go to bed, they're up at 5, and they're ready to go.
They don't want to miss anything.
They don't miss anything. Yeah.
And excessive sleep can be bad.
Excessive sleep's not good either, right? I mean, no, right. So there's a balancing act, and each person has sort of its own little set point. And you probably know when you haven't had enough sleep and you need more. And you probably realize when your body's so pooped out that it's sleeping more than usual. So we all have our norm, right? But what I'm saying is that during an 8-hour sleep, you might have 1, 2, 3, 4, 5 REM periods. And not only would you have 5 REM periods, but each REM gets longer. So if you're just having those 2-hour cat naps, you might get 1 REM cycle, but it's not a very long one. So by having a longer sleep, you're going to get more and get longer REM periods. So those are those memory storing times and those energy restoring times. So that's sleep. Just know your waves, alpha, beta, delta, theta, and recognize the REM sleep story, that's a time of restoration.
Is the theta, is that in between the stages too?
Theta was where? Theta was around 3, right? As you're going into a deep sleep, not the deepest sleep, but in that intermediate, that's theta. Again, it's not in alphabetical order. I wish it was. Okay. Okay, so let's talk about cognition. Another thing that requires many parts of your brain to work in a coordinated way. We might say this is the collection of your reasoning, your perceptions, your intelligence, your imagination, judgment. All of those things are thrown into this thing called cognition. Now, is there one area that does this? No. I mean, a vast majority of the brain is somehow involved in one or more of these areas of your overall well-being. Now, we can study regions of the brain, as we are doing in lab. I mean, we haven't talked about it a lot, but I have given you that form. I said, here, go learn these regions. You know your lobes. You know some of these different regions. And we know this largely through people who have had strokes and individuals who have had localized brain damage. So someone has a stroke, and then we do a PET scan. Now, what does a PET scan do? Remember PET scan? PET scan was a way of imaging using a radioactive glucose type tracer, and cells that are actively dividing would take up lots of the glucose and therefore lots of radiation, and cells that are dead or tissue that's dead would not take up the radiation. So we can localize very specifically where the brain damage is. And then we look at the patients and we say, okay, they just have damage to the right hemisphere in this little area, and what are they doing differently than what they did before? So we started mapping all these different functional regions of the brain, figuring out, okay, if someone has damage to their parietal lobe, then they're going to have problems with perceiving stimuli, right? Now, we know that what important structure is in the parietal lobe? The sensory strip, right? It's perceiving, it's receiving signals. So we know that that post-central gyrus, aka sensory strip, is in the parietal lobe. Some people, if they have damage to a large part of their parietal lobe, will be completely unaware of objects on the other side. They cannot perceive the objects on the other side. Neurology is really fun. Every story that's fun to learn about is the unfortunate consequence of someone's horrible situation. But the stories can be rather entertaining or really more awe-inspiring about, oh, wow, if we do that to the brain, look what happens. The temporal lobe. If you have damage to your temporal lobe, then you're going to have problems with seeing. Perhaps you may have agnosia. Agnosia is the inability to recognize, name familiar objects. So a person with agnosia can no longer, they know what a fork is, but now the next time they see a fork, they can't tell you what it is. They can't go to a toolkit and recognize a hammer. They no longer can understand or name things that they would have had no trouble with beforehand. And that can even go on into recognizing people's faces. Okay? No. Do they get it back? Typically not. It can get better, but typically not. And then in your frontal lobe, When I say frontal lobe, I want you thinking about your personality levels. A lot of your personality is from your frontal lobe. It's also where you have your motor strip. And so it's where you're going to initiate your voluntary movements. But a person who has damage to the frontal lobe may start acting in very peculiar ways, may start doing things that they wouldn't normally do. When grandma starts gyrating in the front window, something's not right, right?
Major injury, like a car accident with a full-on frontal lobe. I was just reading this.
Yep, it could be a closed brain injury, like a TBI, a traumatic brain injury, or it could be a stroke, either one, right? But damage to the frontal lobe could exhibit some of these. Now, it depends upon how much of the frontal lobe and what parts of it. If it's way up here in the frontal lobe, that would be the executive motor or the executive thinking. If it's further back, then it could affect the what? What's at the back end? Yeah, the back end of your frontal lobe is the motor strip, isn't it? So if it was up toward the front, it would be some of your executive thinking and your logic and your inhibition areas. And if you affected your frontal lobe further back and it touched on your motor strip, now you would have paralysis. You would have inability to move certain parts of your body. Remember that little homunculus man? That little guy that stretched out along the motor strip? And so if you damage certain parts of the motor strip, you would have damage or inability to move certain parts of your body. Okay, so a little bit about lobes and cognition. Memory. I've already mentioned some of this. What part of the brain is largely involved with memory? The system is called the limbic system, and within the limbic system, there's a structure called the hippocampus. Memory is not only making new memories, but also learning what we're doing right now. So you're acquiring new information, you're processing that information, and you're somehow going to, in the future, hopefully retrieve that information on the test. And it will be a while before you do the next thing, which is to forget it, right? But forgetting can be as important as remembering. You got to empty out the filing cabinet every once in a while, right? So we have to make, there's only so much we can hold on to. That's not only in our life, but that's also in our memories. That's even within a 2-hour lecture, right? Some of you are holding really well onto something I said 25 minutes ago, and you've already had to let it go. It's gone. It's out of that short-term memory, and you just made room for this new idea. And it's not until you go back and interact with this lecture maybe again on YouTube, or when you go back and look through your Connect assignments, that you'll start to pull all of this back together again. Now, what is amnesia? The inability to remember or to describe past events. Now, there can be either retrograde or anterograde. Now, we've already seen those words. Anterograde was when you were going from the soma down the axon toward the terminus. So that means going what? Going forward, if you will, new information. So a person who has anterograde amnesia would not be able to follow along and make new memories, although they may be okay with their older memories. A person who has retrograde amnesia is a person who might be okay with remembering what's happening right now, but they can't remember something in the past. And there can be combinations of the two. A retrograde amnesia is okay if it involves not remembering the accident, right, as long as they don't lose too much along the way. So sometimes retrograde's a good thing, right? We don't want to remember that traumatic thing that happened in our life. Sometimes that's a gift, to have some little bit of retrograde amnesia. Now, amnesia is typically described as being declarative. Okay? So amnesia, unable to articulate, unable to tell you what happened to me in the past. And you can also, though, have procedural amnesia, which would be the inability to remember how to do something, how to tie your shoes, how to ride the bicycle, that kind of thing. I've already mentioned the hippocampus, a very important part of your limbic system for the forming of new memories. Now, there's a slight difference here, or a slight caveat. It forms your memories. It does not store them. So the hippocampus is knitting them together, but it does not store them. Okay. So the hippocampus is also organizing all this incoming information, right? It's trying to knit together or solidify or synthesize all that's coming in. And we would say that it is memory consolidation. What does that mean to you? What does consolidation mean to you? Trying to put it all together, right? So the hippocampus, is like grandma knitting or pulling things together, and or maybe someone who likes to scrapbook, right? Pulling all the things together into some coherent story or making something that's really cool. And then after that, once we make those memories in the hippocampus, then they are stored elsewhere within the cerebral cortex. We know, for example, that vocabulary and memory of familiar faces is stored in the superior temporal lobe. Okay, I want you to go back. Where did I say about the temporal lobe a while back? It was the temporal lobe where you could have damage to it and not be able to identify people's faces, right? So, or identify familiar objects. So it makes sense that your ability to recognize faces and the storage of faces is involved with the temporal lobe. Your planning, your social roles are in your prefrontal cortex, and that's just a fancy way of saying in the front of in the anterior section of your frontal lobe. So the prefrontal cortex is your frontal lobe in front of your motor strip. Okay, does that make sense? Your motor strip is toward the back of your frontal lobe, and so everything in front of that is considered the prefrontal cortex. And that's where your planning, executive thinking, inhibitions, your roles in the social realm, all that's there. Cerebellum, if I say cerebellum, hopefully you're thinking, oh, that's an area where I am having fluidity of motion, right? It's also part of movement, not voluntary. It's helping you learn motor skills. So I know I said last semester, when I see someone who's just a skilled athlete or dancer, and I watch them, and I'm like, wow, right? I mean, that took a lot of work for them to get to that level of expertise, but they also have a beautiful cerebellum, right? Because they are able to interact, and their motion is so fluid and just a neat thing to watch. So not only is the cerebellum helping you to coordinate your movement, but it's also storing your movement memory. And then finally, the amygdala. I've already mentioned the amygdala was part of your emotional part of the limbic system, and it's also there that you store your memories for emotional things. So someone has a stroke, damage to their amygdala. What do you expect their personality to be like?
They don't really have it.
They may not have any. They may be very, right? They may not have the ability to get excited or to be sad. They may have no real emotional differences because they've lost that part of their brain. One of the classic examples of when, and I don't know exactly why this guy got famous. Maybe it was just so gross an injury. Phineas Gage. Every psych book, every neuro book has this guy's story. And the idea was that, and this was, I want to say, back in late 1800s. And I may be off on that, but I think it's the late 1800s, when they were building a lot of railroads. And this guy somehow got this railroad rod, metal rod, stuck, as you see here, and you would think death, right? Especially back in a time when infection and inability to clean out a wound like this would not have been good. And what they noticed is that this guy started having some really strange personality modifications. And what lobe did we just affect? We just went right through the frontal lobe. Especially we went early in the frontal lobe. We're in the prefrontal cortex. And this guy started exhibiting some really gross behaviors. Very angry, very agitated. He apparently was a really nice guy before all this. And he became very fitful and irreverent and I guess also quite profane. So we realized then from that example that, okay, this part of the brain must have something to do with these higher cognitive functioning ideas, okay, as well as your moral judgment. Should you do it or should you not do it, right? Sort of your inhibition areas. And what do you know from a conversation with me last semester about the frontal lobe? It's also the last part of the brain to become fully developed and especially fully myelinated. And so, your areas of inhibition are not completely formulated until as much as age 25. And by age 30, the brain starts to have negative effects. So there's about 5 good years in there when your brain's fully formed and you're not starting to have some decline. Okay, so again, we're going through these higher functions. Emotions, again, we're talking about a lot of areas of your brain involved with emotions, from your prefrontal cortex, where are we? Up here, to your diencephalon. Next to that, you would want to put what? Limbic, right? The limbic system, all about emotions, and that's also part of or within the diencephalon. So again, we've already said this. The prefrontal cortex is where you have judgment and control over your expressions and also interacting with your emotions. Your feelings of love and fear are a combination of your amygdala, your hypothalamus, and your frontal lobes. Again, just a couple more things about the amygdala. It's receiving information. It's receiving sensory information. It has a role not only in fear, but also in food intake, sexual behavior, and drawing attention to new things. And some of this information goes from the amygdala to other parts of your body. Can your emotions influence your heart rate? Can your emotions influence your blood pressure? Can you be so upset by something that you vomit? Okay? So the amygdala, it's receiving all this sensory information, and your areas of fear and tremendous emotional centers then go on and influence other parts of your brain such that you can physically get nauseous, your heart rate can go up, your breathing rate can go up. I mean, all the things that we think about as being fearful can certainly occur as being influenced from the amygdala. The vagus nerve is definitely part of this. Good connection. So the vagus nerve is the one that goes down and helps to influence your blood pressure and your respiratory rate and your gut. So it would all be connected. Good idea. Good. Okay. And also, like I said, the amygdala is involved with influencing your ability to love or be angry or be fearful or all of those other good and bad emotions. Now, there can also be some shaping that goes on here. And maybe you know someone who's always angry, or someone who, there's something that kind of gets hardwired into somebody. And we know that some of our behaviors in some individuals are learned, right? Our behaviors, not all of them, the nature-nurture thing, but some of our behaviors are learned because of continual stimuli, and we respond to those in ways that some people would consider normal, and then talk to somebody with PTSD, and they'll tell you, okay, now a normal stimulus that wouldn't affect anybody else in the room, now their brain has been wired to overstimulate when they hear a crash, or when they see fire, or when a book slams on the ground and they think it's a gunshot or a mortar shell or something, and they respond in a very different way. So that's all the interaction here of the amygdala, the frontal cortex, your limbic system, all that going on, as well as reward and punishment, right? Part of that's learned as well. So some people respond to things out of a sense of gratification or aversion away from it, and it takes a lot to undo these hardwirings. So once those connections are made, and once those, what's that word we say when things are made easier? Potentiation. Once that potentiation is laid down and those pathways are established, these are very difficult to undo. Not impossible, but it takes a lot of work. A lot of work. Okay. Sensation. We'll finish this up. So where else do we have in the brain? Where do we receive information? When I say the parietal lobe, I hope you're thinking sensation, right? So we say this is your primary sensory cortex. This is along the sensory strip. And we see that sensory strip, right? Now, this is showing you both the post and the pre-central. The darker purple, the one that's more inferior or posterior on this image, is in the parietal lobe, and that's your sensory strip. We know that one. Now, lastly, the special senses. This is exactly what we're doing in lecture, so I can go through this quickly. You know this. You know what parts of your brain your special senses are associated with. So if I say vision, you're going to say, vision is back in the occipital lobe, and the part of the occipital lobe, we would call it the visual cortex, right? The visual cortex is contained within the occipital lobe. Whereas, but it's not all it does, right? Because look what else we have for vision down here. If it's just regular old vision, yeah, that's the occipital lobe. But if we're talking about visual recognition of your faces, right, that was what? That was back in the temporal lobe. Okay, so we don't want to say it's just, all vision is in the occipital lobe, but a large part of it is. Hearing. Hearing, when you see hearing, you want to think temporal lobe and insula. That makes sense, right? Temporal lobe on the side by the ear, that one just kind of makes sense. Also, the insula has some hearing centers associated with it as well. Equilibrium. Balance. We know that's from the what? Semicircular canals, from the structures of the vestibular system. And what part of the brain is doing that? Well, that's the cerebellum is a part of that. Some of the brainstem nuclei, like the basal nuclei and that motor control, is also going on there. Taste and smell. If I say gustatory or taste, that is in the parietal lobe and a little bit of the insula. Whereas if I say smell, we're talking about the temporal lobe and a little bit of the frontal lobe. Now, those kind of make sense when you think about the location of the olfactory nerve, right? The olfactory nerve is sitting there. It's right close to the frontal lobe and near the temporal lobe as well. And the taste going to the parietal lobe, think sensations, as well as to a little bit of the insula. So here's a slide. You've already seen this in lab. I think you should be pretty good with this. Here's our Broca's area. Here's the Wernicke's area back here. Everything else, you can look at this. You may want to go back and look at this particular slide. It might help you in connection with or in addition to that one brain image from the lab. I think this one might be easier in some ways because it's checking right down all of the senses. Motor control. When I say motor, you're thinking frontal. We get that. Okay, no reason to say anything more about that. And then what else is dealing with motor? There's the basal nuclei again. We've already mentioned it. Also the cerebellum, also involved with that coordination of motor movement and maintaining your tone and all of those things. A person who has a problem with their cerebellum would be described as having ataxia, that is having a clumsy gait and uncoordinated movement. A person who had issues with their basal nucleus might be described as having some sort of dyskinesia. So break that down for me. Dys, bad or difficult, kinetic, movement, right? So some sort of dyskinesia would be some sort of movement issues related to the basal nucleus being defective. Language, we've already seen these two in lab. The two big ones, Wernicke's and Broca's. Broca's is all about movement of the muscles in order for speech to occur. Wernicke's is all about the perception of written and spoken language. They talk with each other, and they're both found typically on what hemisphere? Yeah, they're both typically in the left hemisphere. Okay, so here's the left hemisphere view of the brain. And it makes sense when you think about it, because Broca's is up here in the frontal lobe. So that's the movement, the motor coordination necessary for speech, whereas Wernicke's is back here in the parietal temporal lobe, where we think about hearing and perception and receiving information. It makes sense, right? It's not just memorizing, oh, Broca's. No, where is it? Okay, it makes sense. It's motor. And then the last, one word, and then I'm just going to leave you with the cranial nerves on your own. The aphasia, right? Different types of aphasia. Now, aphasia is the inability to, some sort of language deficit, okay, in general. And aphasia is a lack of language. Now, there can be Broca's aphasia. Well, if you have Broca's aphasia, what's their problem going to be? They can't speak, right? They can't get it out. They can't get it out. They can't make the words. They can hear and understand everything usually quite well, but these patients can't articulate what's going on. Versus other individuals have a Wernicke's aphasia. These are individuals who can cuss you out just fine, but they don't understand the instructions. They're having trouble with perceiving the information because their Wernicke's area is damaged, right? They're having a hard time comprehending written or spoken word. And then there are combinations, don't worry about this one, but I want you to definitely understand Broca's versus Wernicke's aphasia because they're going to have different issues with their language problems. And now the last little bit is your favorite part. In 105, I had given you, back in whatever that chapter was, chapter 14, a bunch of slides on the cranial nerves. Here they are again. The ones that are most important for you, though, are the ones we've been discussing in lab. So I want you to at least go through here and before the lab exam, use this list of cranial nerves as a reminder. I won't ask you their location. I'm not going to ask you to identify them by location, but you should know basically for each of these nerves what's its number, what's its name, and what is it doing. Okay? And if you know what it's doing, then you know what would happen if it were damaged. Okay? And so you can walk through this set of slides. I think I've got one slide per nerve as a good quick review on your cranial nerves. And we'll pick up with a quick cranial nerve review on Tuesday. I'll walk through these really quick, and then we'll be continuing into chapter 16 on the senses, which will continue to pull together some ideas from the nervous system. Have a wonderful long weekend.